for you. So I'll kick it off with a little update on Sea Grant activities, including the Huron, Michigan Predator Diet Study, Salmon Ambassadors, the Great Lakes Angler Diary, and a few new educational resources we have coming out. So this first series of slides is really from Brian Roth's talk. Dr. Roth presented up at the Ludington workshop and had some updates from the diet study that's been going on a couple years now. And I should note first off that this is really preliminary data. There are a lot of stomachs coming in. You might remember me talking about this last year when we started this. Really, it started in Lake Huron in 2016, and the idea came up, well, we're doing this effort on Lake Huron. We're looking at all these different salmon and trout species and walleye. Why not expand it to Lake Michigan? Look at both lakes at the same time and all throughout the year. So it's a huge effort, and it looks like we're going to be able to fund this for the next few years, but when we first came up with this idea, the funding fell through for the first year, but all the partner groups involved were so excited about it that we just went forward and collected the stomachs anyway. So these guts wind up coming into MSU's lab and Dr. Roth's lab, and we just kind of scraped together everything we could to fund some students. Sea Grant funded one intern, MSU Extension funded another intern. We had several people like yourselves and several clubs chip in to fund some student research assistants who went through hundreds of stomachs over the course of the summer. So we managed to get on top of things in 2017 and 2018. We still have a backlog of about 1,000 stomachs from 2018, but hopefully those will be done here in the next few weeks, because the good news is we did get grant funding as of 2019 approved that should carry through to 2021. So there'll be a doctoral student at MSU working on this data. The downfall is we have these guts analyzed, but we don't have a lot of data crunched yet. So some of these numbers might change a little bit. That's why we don't have a lot of hard numbers up here. But this is what was gone through as of the time that Dr. Roth presented up in Ludington. And you notice it's not even species by species, right? We've had this collaborative effort. U.S. Fish and Wildlife Service biotechs have been taking guts. MDNR creel clerks have been collecting stomachs at launches. But there are some species that don't show up as much, right? Brown trout in Lake Michigan, we only had 64. You know, it's hard to come by brown trout, but that's where fishermen who are out, especially early in the year like now, if you're out targeting brown trout, you're not going to get picked up in the creel census, and the biotechs aren't working yet. So now through early June is really a great time for anglers to help pitch in and collect some stomachs, especially fish like brown trout and also steelhead. They have a pretty variable diet, so we'd really like to get some higher numbers for them. So looking at the data we do have, you'll notice on these graphs there's a lot of blue, and blue is alewife. So for Chinook and coho in 2017 in Lake Michigan, it's not really surprised that overwhelmingly those fish were consuming alewife. It's also interesting, though, that when you look at lake trout and steelhead, two fish that we tend to think of as being much more diverse in their diets, well, in 2017 in Lake Michigan, when you look at the data by percent wet weight, it was still overwhelmingly alewife, especially for steelhead. We do see more purple there for the lake trout, that's round goby, and the gobies were especially coming from the southern part of the lake early in the year. And we'll be able to, of course, analyze that in a lot more detail in the future. But you can see that it's overwhelmingly alewife, which is interesting because 2017 wasn't a particularly great year for alewife, if you recall. But they were still showing up. Brown trout are a little bit more diverse. Again, we didn't have huge sample sizes for brown trout, but you see more invertebrates by weight, and you also see more round goby. I should mention, too, that the steelhead, it looks like this little tiny slice of the pie is invertebrates. Well, that's thousands and thousands and thousands of little insects. Some of those were so packed that it took the interns a whole day just to go through one stomach and count all the inverts. They were packed, but by weight, it's pretty small. And of course, fish sometimes eat some odd things. This was from a lake trout stomach in Lake Huron, a Coca-Cola label. Apparently they like Coke. They also found a robin's leg, a bird leg, swallowtail butterflies, spiny water fleas. So the lake trout there, they find what they can. So plans for the upcoming year, is to collect as many stomachs as we can get. Again, your help is appreciated. And we'll continue to present preliminary results as they're available. They're also looking at stable isotopes to kind of ground truth what they're seeing in the stomachs. Stable isotopes from the muscle and the tissue of the fish gives us a better idea of what that fish ate over a long period of time, so it's not just a snapshot in time. And they're also using survey caught fish, and this is from the Grand Traverse Band and the Little Traverse Bay bands. They're providing survey caught fish. Again, that's not from the commercial fishery, that's from their biologists who are out there sampling lake trout, walleye, and other species. And it's really going to help us, because if we're relying on all angler caught fish, we don't know if the spoons you're using, maybe they look like an alewife and the fish are just targeting them because they're already eating alewife. Well, we want to be able to ground truth that with fish from nets as well, so that's an important part of the study. What can you do if you want to help? Of course, collect stomachs, especially early in the year and late in the year, especially brown trout and steelhead. You don't have to collect every single fish. And these stomachs can be dropped in the same freezers that are taking code wire tags. So if you see those freezers at local marinas or cleaning stations, you can use those. We have a bunch of tags back there in the brown boxes if you want to just grab 25. There are lots of 25 or 50 and just fill them out over the course of the year, which is really appreciated. We also still have the crowd power site up. We raised about $6,000 from anglers and groups last year. That was really helpful. So if you want to fund some of this work going for students going through GUTS, that's still up and running. And I should mention too that we've got a lot of ways to reach out to folks. We have a Facebook page that's run by one of the lab techs there, and there's lots of cool gross pictures, so that's nice. Yes?
On the slide before, it said zeros are really important.
Oh yeah.
What are you talking about there?
Well, this is because people have a tendency, let's say you catch a big fat lake trout and you say, oh, that's going to be really interesting. I'm going to take the stomach from that fish. Well, the empty one right next to it is just as important. We can't have people biasing the study by only taking full fish. So what we're asking is, you don't have to take every fish, but if you go out on a trip and let's say you're going to collect a steelhead stomach on that trip, take all the steelhead stomachs from that trip. You don't have to take all the kings, you don't have to take all of every species, but if you're taking fish of one species, take all the fish of that species from that trip. That'll help us to prevent that bias, because one of the things we look at is the percent empty too, so we can't get that without zeros. Yeah.
All the, I would think this is pretty common knowledge, but most of the lake trout come in the boat. They're either spitting up all the alewives or they're already throwing them up as you're pulling them in.
Yeah, there's not much we can do about regurgitated fish.
Well, how do you handle that in your data?
Yeah, there's not really a way that it's possible to handle those fish, unfortunately. Of course, that is going to happen. We just ask people to do their best. If you know a fish regurgitated and there's something coming out of its throat, for example, then you can say, oh, well, I better put that in the bag with the stomach. But if it's in the lake, there's nothing we can do about it, unfortunately. I should mention, though, it's a good point, because you see those big fish. You see the 6-inch alewife in there. But the lab technicians are incredibly thorough. They're going through every little fold, and if they find a little bone, a little otolith in there, they can say that, well, this fish did have a 5-inch alewife in there, a week ago, and this is all that's left. So they can tell a lot from what they get. Yes?
It's the other reason we look at netted fish, because usually trap nets are brought up to a flow they're not eager. Another way to see it.
Because we catch a lot of lake trout, and there's hardly anything in their stomachs. But if they're looking at that detail, maybe
I don't know, if people send me pictures of lake trout stomachs every day. Look what they're eating.
Yeah.
They don't irritate anything.
And another cool thing is, the comparison from lake to lake and from area to area and year to year. There's probably always going to be some level of regurgitation. So the comparisons through time should also hold true. So switching gears here a little bit. Oh yeah, you want more on this diet?
Does everybody here know where the closest drop box is for this port?
Does everyone know? I know we've got it in our packet back there, and I can't tell you off the top of my head where it is, but I think it's at the Black River Launch. There's a cleaning station there, right?
So there's a freezer there?
Yep, there should be a freezer there. There should be a sign. We've got a copy of the sign up there. And
Wolf Lake will take it if you're heading on back to Kelm.
you should be able to drop them off at the launch there at Black River as well. And they get picked up, about every couple weeks by DNR technicians. So switching gears a little bit, we also have the Salmon Ambassadors program that a lot of you have been chipping in to help with, looking at the percent wild Chinook salmon. All the Chinook salmon have had adipose fin clips the last few years. And as you see, we've been doing this since back in 2013, and we have over 10,000 Chinook salmon recorded as part of this program, which is pretty cool. This last year, in 2018, the results were pretty consistent with what we've seen the last several years. Ludington, the numbers dropped down a bit, and the percent wild dropped down a little bit from what we were expecting, but that may have been to low sample size. But usually, we get in the 70s and 80% range in Michigan waters. That's 70 or 80% wild. A little bit lower if you go over to the Wisconsin side. Illinois and Indiana, again, very low sample sizes, but down at 22%. And Northern Lake Huron was about 50-50 wild and stocked fish, and that's kind of gone up and down a lot over the last few years. But Northern Lake Huron, you've got fish returning to the Swan Weir here, and then you've got other fish that are wild that are predominantly coming from Georgian Bay tributaries. So that can really affect things. And we always do this survey at the end of the year. It's interesting to see how people's perception of how good fishing is changing year to year. And again, it's kind of a middle of the road year in 2018. One thing that's really jumped out the last several years, though, is that people tend to find much better fishing in May, June, and then they're a bit disappointed when the end of the year runs come and the fishing just kind of fizzles. We don't really see the great pierhead kind of fisheries that we used to, especially in the southern part of the lake. One thing we've developed in conjunction with that is the Great Lakes Angler Diary, which is just an app. It's been a website that anyone can use with any device, and we've kind of tailored it to be a little bit easier to use. We have new apps coming out for Apple and Android pretty soon, but we're not launching them quite yet. They do work a lot better. They're a lot simpler and hopefully more fun to use. But the plan is to probably phase out the Salmon Ambassadors now that the Chinook salmon are not being coated wire tag. Steelhead have been stocked with all adipose fin clips and coated wire tags since last year. So as those fish start to age up and the unclipped stocked fish start to die off or phase out of the system, we'll start phasing in a Steelhead Ambassadors program. And last thing I wanted to mention is we have a few new educational products, a couple of books that Sea Grant staff have been involved in. It's, first off, the Howard Tanner autobiography. If you don't know Dr. Howard Tanner, he's probably the man most responsible for the salmon fishery we have here in the Great Lakes. He's known as the father of the Great Lakes salmon fishery, and stocked coho salmon first, and then shortly thereafter Chinook salmon. It's a really interesting story that takes you back to the beginning of the fishery and kind of the whole political climate that was going on at the time, and some of the hurdles he faced, and some of the really amazing stories about those first days of coho fever. So if you get a chance to check that out, it's a great read. I also included a little review of the book and some information on how to order it in your packet. Another thing that we put together is the Life of the Lakes. Sea Grant staff, Michigan Sea Grant staff, have gone through 4 revisions now of the Life of the Lakes, because the lakes change so much, every few years we have to rewrite it, right? So we got more stuff in there about the effects of zebra and quagga mussels and round goby, and a couple of neat things that we've been able to do is to kind of help people visualize what's going on, some of these basic ecological principles. One that I worked on a lot was the question of what it takes to make 10 pounds of fish, and Chuck helped me out with this, Chuck Medengian helped review it all and make sure it was on point. But it takes about 75 pounds of alewife to make 10 pounds of salmon. And if you follow that down the food chain, that takes 2,000 pounds of zooplankton to make 75 pounds of alewife, and over 7,500 pounds of phytoplankton to make 2,000 pounds of zooplankton. So you're talking about 4 to 5 tons of plankton going into each 10-pound salmon that you catch. So when we talk about changes in the lower food web and how the mussels have messed up the phytoplankton, and you're seeing that in the zooplankton, this is why it's so important. You screw up the balance a little bit, and all of a sudden you can't produce as many fish at the top of the food chain. So this is just kind of a neat figure. It's, you know, it's hard to compare this, and people have asked, well, can you do that for lake trout? It gets very complicated, and in fact, this really applies before the quagga mussels came in, because the alewife, it's a simple situation with salmon. Salmon eat pretty much all alewife, and before the dry-scented mussels, the alewife were predominantly eating these zooplankton that were phytoplanktivorous. But now things have changed to where alewife are eating more possum shrimp, which are themselves predators. So you also have this inefficiency coming up in the system. So there's also an article on that if you're curious more of the details and kind of how that worked. So that's all I have. If you got any questions, yes?